The materials that are used for the Hopkins-Coles test are the 1% egg albumin, 1% casein, 1% gelatin, 1% tryptophan, Hopkins-Coles reagent, and concentrated sulfuric acid. For the procedure, you mix 10 drops of Hopkins-Coles reagent to 1 ml of the following, which are 1% egg albumin, 1% casein, 1% gelatin, and 1% tryptophan. After that, you add a few drops of concentrated sulfuric acid down the side of the inclined test tube and fix the test tube upright slowly so that a violet ring formed at the junction and it can be clearly seen. The Hopkins-Coles test detects tryptophan, an amino acid in proteins. It reacts with glyoxylic acid and concentrated sulfuric acid, forming a violet ring. The color change indicates the presence of proteins containing tryptophan. Between the 4 chemicals that are used in the experiment, only 3 have positive results and only 1 has negative results. The positive results of the experiment are the 1% egg albumin, 1% casein, and 1% tryptophan, while the 1% gelatin is the only negative result. The chemical reaction in the egg albumin solution shows a dark violet ring in the middle, while the tryptophan solution shows a light yellow ring in the middle, which then turns into dark green at the bottom of the solution after a few minutes. For the casein, there was no reaction at first, but then after a few minutes, a light purple ring starts to form near the bottom of the solution. For the gelatin, since its negative result, there was no reaction that was seen, even after a few minutes.
First step is to prepare all of the materials that is needed, especially the chemicals that is needed for the experiment, and also label every test tube that is needed. For example, in test tube 1, we have the egg albumin, test tube 2, we have the proline, and test tube 3 is the alanine. And also preparing for the chemicals that is also needed, the ninhydrin solution and the egg albumin, and 1% of proline and 1% of alanine. The second step is adding the ninhydrin solution to every test tube. So using a pipette, add 1 ml of 0.2% of ninhydrin solution to. So the purpose of this experiment is to detect the presence of protein or free amino acid in a sample by using an ninhydrin. An ninhydrin reacts with amino groups which characteristic of amino acid and proteins to produce a color change. This reaction is commonly used in biochemical tests to identify the quantity amino acids or protein. And in terms of the chemical elements reacting to it, we have the amino acid or proteins by providing an amino group of, for example, of negative NH2 or negative NH. And in terms of the ninhydrin, reacts with this group releasing CO2 and NH3 and forming a colored complex. So after adding the ninhydrin solution, next we're gonna do is to add the samples. So in test tube 1, add 10 drops of egg albumin, followed by in test tube 2, 10 drops of 1% of proline solution, and lastly in test tube 3, 10 drops of 1% of alanine solution. Lastly, we're gonna place all of the test tube in a water bath and set to boiling temperature and heat it for at least 5 to 10 minutes until it color change. So based from my observation and also the research that I have conducted, in terms of the positive result of ninhydrin solution, it has the appearance of a purple and deep blue color, which indicates a positive result. These are the results in terms of the different
experiment. Right? This is egg albumin.
In terms of the positive result, as you can see, the egg albumin and also the 1% solution of alanine has a positive result because of their color change. In terms of the egg albumin has a deep blue color, while in terms of alanine it's a purple color. So meaning to say it indicates a presence of amino acid, peptides, or protein itself. So in terms of the proline, it doesn't have a color change, so meaning to say it has the absence of amino groups in the sample. So that's all for this video. Thank you all for watching.
So for the Burette test for proteins, specifically peptide bonds, for the materials, we need pipettes for measuring and transferring reagents. We need 3 test tubes, 3 extra small test tubes, and 3 solutions that we are going to test, which is the 1% glycine, 1% gelatin, and 1% albumin. We also need 10% sodium hydroxide and 1% copper sulfate for this test. For the first step, we need to label the 3 test tubes with glycine, gelatin, and egg albumin. As you can see, we are adding 1 ml of each solution to the corresponding test tube. Here we are adding 1 ml of 1% albumin, 1 ml of 1% gelatin, and 1 ml of 1% glycine. We also measured 1 ml of 10% NaOH in a small test tube and then added it to the 3 solutions, which are glycine, albumin, and gelatin. Now, we are going to add 2 drops of 1% copper sulfate to each test tube, which we are going to shake and then observe. For the egg albumin, we can observe a pink color, which is also a reddish pink to violet color, which indicates a positive result. For the gelatin, there is a dark purple reaction, which means there is presence of peptide bonds, so it is also a positive result. For the glycine, we can see that it is only light blue, which indicates a negative result. The Burette test tests the presence of peptide bonds. As you can see, there is no reaction for glycine because glycine is a single free amino acid. As we all know, a group of amino acids are the ones that makes up a peptide chain and is bonded by a peptide bond. And when we have a single amino acid with no other amino acids bonded to it, there is no peptide bond. And Burette test for peptide bond, so therefore the reaction is negative. For the gelatin, we have a purple reaction, which means that it has many peptide bonds. And for the egg albumin, there is a pink color to reddish violet color, which means that it is still positive, but there are minimal chains, but it is still considered as positive result. The color comes from the reaction of the nitrogen atoms in the peptide bonds with the copper in the copper sulfate. And as we can see from the experiment, we added 1 ml of NaOH to each of the 3 solutions. This is to make the solution in an alkaline condition, because making the solution in an alkaline condition is necessary to help the reaction between the copper sulfate and the nitrogen atoms of the peptide bonds. The alkaline conditions makes the copper of the copper sulfate in its reactive state, so that we can make sure that it will react with the nitrogen atoms of the peptide bonds that causes color change.
So in the first test tube, I'm adding 20 drops of egg albumin and in one another test tube, I will add 20 drops of 1% tyrosine. And since we didn't have protein hydrolysate, we disregard that and move to the other reagent which is the 1% casein. I also add 20 drops of it in one test tube. And after that, I add 10 drops of 10% NaOH in each test tube. And after adding 10% of NaOH, I add 5 drops of lead acetate in each test tube. And after adding all of the reagents in the test tube, I shake it and place it in boiling water bath. After a few minutes, the reagents started to change its color. As you can see, the albumin changed color and turned black, and the casein turned transparent yellow, and the tyrosine became clear. This experiment is used to describe the presence of sulfur. The egg albumin contains sulfur, that's why it turns black. However, casein and tyrosine does not contain sulfur as it is remained the same or in same color.
To test the presence of carbonates in the urine, a reaction with acid can be done because carbonates react with the acid to release carbon dioxide. So the presence of bubbles or air indicates presence of carbonates. In this test, we will use hydrochloric acid in fresh urine samples. We are going to measure 5 ml of fresh urine sample and label it. Then, we are going to add 20 drops of hydrochloric acid and observe for the reaction. As you can see, there are no bubbles in the urine, so it is a negative for carbonates. The result is a good result because carbonates in the urine could be because of UTI or bacterial infection. It could be because the blood is too alkaline, which is bad, or other health condition problems.
Test of creatinine. The materials that are used are 1 ml of saturated picric acid, 1 ml of 10% NaOH, and 5 ml of urine. In a test tube, add 1 ml of saturated picric acid and 1 ml of 10% NaOH to the 5 ml of urine. After that, observe the color that is formed. The color that is formed is a red-orange color, which is the positive result, and it occurred because the creatinine in the sample reacts with specific reagents such as picric acid in an alkaline solution, forming a colored complex that indicates its presence and concentration. The purpose of the creatinine test is to evaluate kidney function by measuring the levels of creatinine in urine, which can also help detect abnormalities in the presence or concentration of specific elements or compounds in the urine. The red-orange result in the creatinine test typically indicates poor health and suggests potential kidney dysfunction or damage.
In terms of the test of sulfate in the urine experiment, the first thing we have to do is prepare the materials. So, the materials that are needed are test tube, 10% of hydrochloric acid, 5% of barium chloride, urine sample, and dropper. So, next to our procedure is to add the samples. So, using a clean test tube, we're gonna add 1 ml of 10% of HCl to it using a dropper. Then, using another dropper, adding the 5 ml of urine into it and mix it well. In terms of the purpose of this experiment, is to indicate the presence of sulfate ions in the urine. By adding hydrochloric acid and barium chloride, a chemical reaction occurs that produce a white precipitate if sulfate ion are present. This test helps to determine whether the sulfate, which are byproducts of protein metabolism, are excreted in a normal or abnormal amounts in the urine. In terms of the specific reaction occurred in the experiment, when barium chloride BaCl2 is added to the urine that contains sulfate ions SO4, raised to negative. The following reaction are BaCl2 plus SO4, and the product will be BaSO4 white precipitate plus 2Cl. So, in terms of explaining this, the in the urine react with barium ions from BaCl to barium sulfate, which is a white insoluble compound. Then followed by adding few drops of 5% of BaCl Cl2 or barium chloride. So in terms of this experiment, I uses 9 drops of BaCl, so that it has a much more of a white precipitate result. As I have said before, in terms of the positive result of the experiment, the is the formation of a white precipitate after adding BaCl, and this precipitate is a barium sulfate, BaSO4, which insoluble in water and confirms the presence of sulfate ions in the urine, while hydrochloric acid ensures the reaction is free of other ions like carbonate that might interfere as HCl dissolve unwanted precipitate like barium carbonate. The result of the experiment, whether it's good or bad to our body, it indicates that, or a white precipitate in the experiment typically reflects on the normal metabolic process unless it is excessively large or completely absent, in which case it could signal a potential health concern that requires a further investigation.
Test for bile acids for the urinalysis. So the materials needed for this test is a pipette, test tube, beaker, urine sample, and powdered sulfur. So in the experiment, first you're gonna need to get 5 ml of urine sample, as can be seen in the video. I'm measuring the urine with a pipette in order to get a 5 ml of urine into the test tube. This is so that we will have much more accurate results and easier easier experiment all throughout. Then after getting the 5 ml of urine sample, we're now gonna put it in a water bath, which is in a beaker, and then we're gonna leave it there for 30 minutes. After 30 minutes have passed, we're now gonna put a pinch of powdered sulfur in the test tube, and if the powdered sulfur didn't sink like can be seen in the experiment, so the result was negative. That is good because it means that there's no liver or potential liver diseases.
In urine, we test for protein. In one test tube, I add 5 ml of urine sample. And after that, I add 1 ml of 5% acetic acid along with saturated NaCl solution. Then I repeat those steps and put 5 ml of urine sample on one large test tube along with the 1 ml 5% acetic acid and 3 ml of saturated NaCl solution, but this time I put 5 ml of albumin. Then I heat the upper portion of the test tube over the flame until I saw a reaction. And since my sample 1 doesn't have any reaction, I move to my second sample, which is the urine sample but with egg albumin. You can see the second sample has a positive reaction because of the egg albumin. This means that the urine has a protein. And here's the comparison of the two samples. The clear one is negative, meaning it has no protein, and the other test tube that has formed and turned cloudy is positive, meaning it has protein.
So what is the purpose of the experiment, or how can it detect the presence of specific elements in the urine? The purpose of the Benedict's test is to detect the presence of the reducing sugars, specifically glucose. And reducing sugars such as glucose can be found in urine when blood sugar levels are high, such as in people with diabetes. The test helps identifying elevated glucose levels that may indicate potential health issues. So, what is the positive result of the experiment? A positive result in Benedict's test is indicated by a color change, which is yung green nga, nag-green yung sample urine namin. Yung sa experiment namin, the positive result was the appearance of green color with a little, no, with precipitation talaga siya, which indicates the presence of small amount of reducing sugar in the urine sample. So, yung reaction naman ng experiment, in the Benedict's test, the Benedict reagent contains copper sulfate, which is yung blue, and ginap namin in 5 ml. When reducing sugars are present in the sample, they reduce the copper ions in the Benedict's reagent to copper ions, which is this reduction reaction results in the formation of a precipitate of copper oxide, which can appear as green, yellow, orange, or red color, pero sa amin is na green, depending on the concentration of a glucose in the urine. A green color indicates small amount of reducing sugar, while higher concentrations would show yellow, orange, or red precipitates. Is the result of your experiment an indicator of good or bad health? If it's bad, meron bang parang malfunction in the body nung may-ari ng urine? Well, a positive result in Benedict's test indicating the presence of glucose can be indication of a poor health, particularly if the glucose is present in the urine at significant levels. Pero kasi yung sa amin, we indicated to the professor na yung nagbigay ng sample ng urine is nag-inom last night. So which made it na parang because of that, nag-positive yung result. So this could suggest a malfunction in the body such as diabetes or impaired kidney function. But I don't think talaga na parang unhealthy yung nagbigay ng. I think nagsumakto lang na she drank alcohol the night before that. Kaya nag-positive yung result. Sa healthy individual naman, glucose is typically reabsorbed by the kidneys and no glucose should appear in the urine. When blood sugar levels are too high, which is called hyperglycemia, the kidneys cannot reabsorb all the glucose, leading to presence in the urine.
Rotera's test. So, place 5 ml of urine in the test tube and then add 3 drops of 5% of NH4 to SO4 and also 5% of sodium nitroprusside. After that, carefully add 1 ml of concentrated NH4OH and set aside for 2 to 3 minutes before observing. After setting aside that, repeat steps 1, 2, 3, but do not forget to add the 5 drops of acetone to the urine sample. So this is the second sample of test for ketones. So 5 ml of urine and then 5 drops of 3 drops of 5% NH4 to SO4 and 5% of sodium nitrogen.
Thank you. Ayun, ano yan? So, thank you. Ketone ba yan? Ano yung sample ko na nangyari? Sample ko yun. May acetone dito. Pwede ba yan? Ilang ml? Ilang gram?
So 5 drops of acetone to the urine sample. So in the first test, the test for ketones is negative, and then it came in the second test, it came as positive because it has acetone.